Today, a breakthrough lands in your chat. SandboxAQ's cutting-edge drug discovery models are now embedded directly inside Claude, ready to run with a single prompt. No installing, no GPU farm, just type and watch molecules appear in seconds, as you'll see in this quick screen record right here, right now for anyone can. The screen flashes with rapid cuts, molecules spinning, bonds forming, data streams flowing, all set to a pulse-pounding beat. On-screen text pops up in bold, kinetic typography, from idea to candidate in seconds. Every frame screams that the future of drug design is here, and it's moving faster than ever for you. Now imagine harnessing that power yourself. No PhD, no lab coat, just curiosity and a keyboard. What if you could design drugs without a PhD? The answer is already inside Claude, waiting for your first prompt. Let's dive in and see what you can create. Stay tuned for a live demo that shows exactly how simple it really is. Think of AQChem as a master chemist who can instantly sketch how a drug molecule will fit into a disease protein, like a key slipping into a lock. It evaluates millions of possible shapes in seconds, predicting binding strength with lab-grade precision, all without ever touching a test tube. AQBio works like a seasoned biologist reading the cell's inner dialogue. It models how proteins interact, fold, and signal, flagging which targets are truly druggable. By simulating these biological conversations, it cuts through noise and highlights the most promising therapeutic avenues. Compared to traditional screening that can take months and cost millions, SandboxAQ's models deliver comparable, or better, accuracy in a fraction of the time. Early benchmark studies show a threefold increase in hit rate while slashing compute cycles from weeks to minutes. The breakthrough lies in democratizing this power. Researchers, startup founders, or curious enthusiasts can now invoke AQChem and AQBio through a simple prompt in Claude. No PhD in computational chemistry required, just type, get results, and iterate faster than ever before. First, open Claude and look for the prompt library icon in the sidebar, usually a bracket or slash menu. Click it, scroll to the SandboxAQ section, and you'll see ready-to-use prompts for AQChem and AQBio. No installation, no config, they're just a click away, ready to turn your chat into a virtual bench. To run a prediction, simply type the slash command for the model you need. For AQChem, type /achem predict and press enter. Claude will respond with a form asking for your molecule's SMILES string and the target protein sequence. Fill in the fields, hit submit, and the model starts working instantly, all inside the same chat window. After you submit, Claude displays the results, a predicted binding affinity score, a 2D depiction of the molecule, and suggested optimizations. You can tweak the SMILES directly in the chat, change a functional group, resubmit, and watch the score update in real time. It's a conversational loop that feels like chatting with a lab partner who instantly returns data. When you're satisfied, click the Export button that appears below the output to download the SMILES, the protein sequence, and the prediction report as a CSV or JSON file. No code, no terminal, just a few clicks and you have a lab-ready dataset ready for synthesis or further analysis. That's how Claude becomes your personal lab assistant, turning chat into discovery. We start with a clear disease target, the KRAS G12C mutant, a notorious driver in lung cancer. In Claude we type a prompt, design a small molecule inhibitor for KRAS G12C, high selectivity, oral bioavailability. Claude pulls up SandboxAQ's AQChem model, acknowledges the target, and asks for any constraints, like less than 500 DA or no reactive groups. This simple ask sets the stage for an AI-driven hit hunt. Within seconds Claude returns 5 candidate SMILES strings, each paired with a predicted binding affinity, 9.2 to 10.1 kcal/mol, and a quick 2D sketch. Compare that to a traditional high-throughput screen that would take weeks to assay thousands of compounds. Here we have AI-generated hits instantly, and the interface lets us click any candidate to see a 3D docking view rendered on the fly. We pick the top hit and ask Claude for optimization, improve solubility while keeping affinity above 9.5 kcal/mol. Claude runs AQChem's iterative redesign, proposes 3 analogues with modified side chains, and supplies new predicted solubility values, plus 0.8 logs. In less than a minute we have an optimized lead ready for synthesis, a process that would normally require multiple rounds of medicinal chemistry. Finally, we request a quick ADMET snapshot. Claude calls AQBio, returns predicted metabolic stability, HERG liability, and blood-brain barrier permeability, each color-coded for risk. The whole workflow, from target definition to ADMET profile, takes under 2 minutes, versus months of wet lab assays. The screen splits to show a timer, emphasizing the dramatic time savings. To wrap up, we hit the export button. Claude bundles the final smiles, docking poses, and ADMET report into a tidy zip file ready for the synthesis team. In total, we moved from a disease concept to a vetted hit candidate in under 3 minutes, an order of magnitude speedup that reshapes how fast drug discovery can happen. SandboxAQ inside Claude drops the lab door wide open. You get research-grade molecular predictions without a GPU cluster, no code, and no PhD. The result is speed, hits appear in seconds, plus a dramatically lower barrier to entry, letting anyone with a laptop join the discovery race. In short, accessibility, velocity, and democratization are now a single prompt away. Ready to test it yourself? Head over to the SandboxAQ Claude portal, hit the Start Free button, and claim your no-cost tier. The page walks you through linking Claude, selecting a model, and running your first prompt. No sign-up hassle, no credit card. Within minutes you'll be generating candidate molecules and seeing real-time scores. Join the growing community of innovators, post your results, ask questions, and share tips on the official forum and Discord. Collaboration fuels faster breakthroughs, and the community roadmap already promises new target libraries and batch mode tools. Dive in, experiment, and help shape the next wave of AI-driven drug discovery. Imagine the future, every researcher, startup, or curious mind able to prototype therapeutics in minutes, accelerating cures and reducing costs globally. With SandboxAQ and Claude, that future starts now, so type that first prompt, watch the chemistry unfold, and be part of the AI-powered drug revolution.